Good afternoon, everyone. For once, we have a relatively short lecture, so there's a good chance we'll not be in here till the exact end of class today. So just as always, remember that the quiz for this week is due at our normal time. Y'all should know the drill by now at this point. Do remember that you get 2 of them to drop, but honestly, unless it's like the exact last 2 quizzes, I wouldn't just stop taking them. It's taking me a little bit longer to get tests back. So expect maybe Tuesday or Wednesday instead of Monday for me to have everything finalized. Yes. No, it'll automatically do it on web courses for the quizzes. Also, don't forget that the Connecting with Biology Q is up as well. So I've seen a decent number of people do it, and they've seen some really cool stuff. So seems like people are having at least a little bit of fun with it. I've saw like a ton of gopher tortoise observations, which those are always kind of fun. But do remember, if you want your best chance at seeing, especially a lot more on like if you don't want to have to just look for bugs for your animals, go out in the morning or in the evening, like right around sunset or right around sunrise. Like it's going to be really pretty out there. It'll be nice and cool, so it's not stupid hot outside, and you can see some really cool stuff. Also keep in mind too that like last time, I'm going to be offering extra credit for exam 3. All the instructions for that should be up on web courses already, so just take a look at that. All right. So Wednesday, we were talking about mitosis and why it's needed and all that kind of fun stuff. But today we're going to take kind of a quick side trip and look at meiosis specifically, because meiosis is different enough from mitosis that it's important to highlight. And it has a very, very specific and important part of the biological process, right? In order to have, healthy sexual reproduction, you have to have this process work correctly. Now, as we mentioned, especially last unit, asexual reproduction is extremely rare. And this is pretty much isolated to single-cell organisms that are going to primarily reproduce asexually. But among multicellular organisms, it's not common at all. So whether that be plants, whether that be animals, you just don't usually see asexual reproduction. Now, I do want to highlight that there are a couple of examples of this that we've kind of talked about. You've heard my creature killer story of that wonderful cottonmouth. Ball pythons especially can also do this parthenogenic birth, where it's that clonal version of it. And do remember that, like, even when you have these circumstances, maybe 1 out of the 10 offspring are going to survive, and even then it's not necessarily a super healthy animal. Up in the corner there is one of what they call the unisexual salamanders. Really funky organisms. They have like 3 copies of their genome. So instead of having 2 or 4, like a normal organism that you would associate with, any of those odd numbered things cause us to have some really weird both reproductive behaviors as well as just general genetics overall. So they're really cool. There's entire stories on them. I'll have to see if I can post a video about it at some point. And finally, these weird whip-tailed lizards from out west that are pretty much entirely female populations, where they've just completely gone without having the ability to, or having the need for the male sex, and just kind of gone with it. Now, like we talked about last time, mitosis has a couple different roles, right? It's obviously underlies all of asexual reproduction, because you're going to be generating that perfectly genetically identical offspring. And again, you can see this in a classic example of something like an amoeba, which is a little protist that's going to be a single-celled organism that's reproducing on its own. However, it also has a lot of benefits as well in regards to, eukaryotic organisms that do rely on sexual reproduction. They do still use mitosis for other reasons. Now, meiosis itself is required specifically for sexual reproduction. This is the type of cell division that generates your typical egg and sperm cells. And keep in mind too that these are vastly different depending on what things you're talking about, whether you're talking about pollen in trees or the seed ovary in the bottom of like a flower, something like that, versus humans, which have a lot of different shapes and structures. But in general, just think of it as a broad spectrum approach of just calling the biological male sperm and the biological female egg. Now, each of these specialized sex cells lies at the heart of sexual reproduction, because ultimately what has to happen in order for sexual reproduction to work and for you to have that proper exchange of genetic material, you needed to cut down like about half of the chromosomes. So that way, when you combine them back together, there's not too much genetic material and you make mistakes. Ultimately, this genetic diversity is worth it, right? So going through the process of attracting a mate and producing sex cells requires a lot of energy and is costly to the organism, right? Because honestly, if you're not using your sperm cells for reproduction, they're just waste, right? That's ultimately, they're just junk, leftover energy that your body's putting into making something you don't need. However, because of it is worth that cost though, because it can create and maintain this genetic diversity. Now, as we've kind of talked about, so remember, these are sister chromatids. So these by themselves are paired up and have the exact same copies of each other. The homologous chromosomes are a little bit different. Basically, it means that this is a pair of chromosomes that have the exact same size and structure, contain the same exact genes, but they may not be the same copies of the said gene. So for instance, for the FOX2P gene that we were talking about last Wednesday, you may have two versions of that. You may have an A and a B version, where you could just have both A's or both B's. It just kind of depends. So this is what allows you to have those different versions of that, or a different version of that gene. And what we call those different versions of a gene, it's called an allele. Now, each of these come in a pair, where one chromosome will come from the biological mother and one will come from the biological father. Now, keep in mind too that this can get really complicated when, especially factor in things like there's a lot of plants as well as a lot of kind of, what is the term? Invertebrate animals that rely almost exclusively on having both sexes present in a single organism. And we'll talk more about like the actual terminology for that. But basically, what that allows them to do is, even though they may be the biological father for one thing, they might be the biological mother for the other. Just kind of depends. Now, diploid cells, which means simply there's two sets of homologous chromosomes. Di2, right? Now, it makes it kind of obnoxious that they call haploid for one pair, because if you think about it, it should be like uniploid or something like that, but it is what it is. Ultimately, sexually reproducing organisms are going to consist mostly of diploid cells, which contain two full sets of chromosomes here. So each of these is a homologous set of chromosomes. They have not gone through the duplication process yet, so there's no extra sister chromosomes here. But for instance, you have 23 total pairs in humans, with that last pair 23, being your either your XX or your XY chromosome. Now, one full set of these chromosomes is inherited from each parent. So when you map out all of these different chromosomes, this is what you call a karyotype. So the image to the right shows you all 46 chromosomes from a diploid human cell. Again, they're not grouped, or they're not in that sister chromatid form, they're just in a single block by itself. And those 46 chromosomes have been arranged into 23 pairs. Now, 22 pairs of these are what we call homologous autosomes. It means they're relatively the same, they share the exact same structure, and they share the exact same number of genes. However, for that 23rd pair, that's going to consist of those sex chromosomes. Now, in mammals, that's where you get that XY and the XX, where XX for biological females, XY for biological males. Now, the reason why we point this out specifically is if you look, here's your traditional XX gene, right? Here's your XY gene. Notice how much smaller that Y gene or that Y chromosome is. And it's because it's missing a lot of the same genes that you find on the X chromosome. And as a result, it can cause some very weird pairings when it comes to, say, for instance, there are certain diseases that can be a carrier for mom, and mom will only ever get it if she's completely double recessive, which means she has both of those copies at her gene. For that specific, she'll have both copies of that allele at the gene. Whereas in this case, the biological male would automatically have it just if mom gave it to her. And we'll talk about how to break that up and how to determine those situations a little bit later. Now, these homologous chromosomes can carry the same set of genes, right? So the homologous chromosomes are not identical. The nucleotide is not the same. However, they do carry the same coding mechanism. So it's kind of like a folder in your desktop, right? You might have two versions of this paper that you're writing, but one is from a lot more recent than the other. And so that change in difference can cause differences to happen. Now, each chromosome in a homologous pair has its genes from the same trait, and the genes are arranged in the same order. And that's really important. But again, these homologous chromosomes can carry different alleles. So recall that allele is a version of the gene that encodes a different version of the same protein. So going back to that translation step, right? There might be just a slight difference in the nucleotides that's causing maybe a single swap out of an amino acid somewhere. And that's different enough that it changes everything. Now, every person is going to inherit one allele per gene from each parent. So you'll have an A from mom and an A from dad, or an A from mom and a B from dad. It all just kind of depends. For example, here, this A and the D alleles are from each parent are the same, while the B alleles are different. So see, in this case, you've got gene A, gene B, and gene D. Now, up on gene A, both of them have the exact same alleles present. On gene B, they're different here, where you've got one having big B, and the other one having little b, and then finally when you look down at here, they're both the same again as well. And again, note this is the replicated chromosome. You have your two sister chromatids, and then you have your homologous chromosomes. You'll want to make sure that you keep those things separate because they are different. All right. So on this image below, it shows two chromosomes from one of the goat's body cells, which statement is possibly true. A, both came from the mother's egg cell. B, the chromosomes are not homologous because they have different genes. And finally, C, one is identical to two. And then D, 1, 2, 3, and 4 are identical. All right. So let's kind of talk through this here for a second. Why is A wrong? Yeah, exactly. Pretty straightforward. One is coming from mom and one is coming from dad. These chromosomes are not homologous because they have different alleles. I don't know, those look like the same alleles or those same genes to me, right? So they're homologous because they have the same genes, not because they have the same alleles. So that's incorrect. C, one is identical to two. What are these called? Sister chromatids, right? What are sister chromatids? Identical. So in this case, the answer is C, 1 and 2. Now, there is a possibility that D could be correct, but we don't know for certain because we don't know the information about what alleles are present, right? So this could be, it's the same gene, right? But what happens if it's big A, little a, little a? That does, if we don't have that information, so we can't make that judgment yet. Yeah.
So what's the difference between the gene and the allele?
So a gene is a specific thing that's being coded for. And an allele is a different version of that gene. So you have version A and you have version B.
So would the gene be like capital A, and the allele would be capital A?
Yeah, exactly. So the gene itself would be, it doesn't have to be both big A, big A.
Yeah.
In that case, it could be big A or little a, whereas the allele would be in that specific organism, that one has an A versus in another organism has a little a. Yes. We don't. And so that's why we know that 1 and 2 are identical because they're sister chromatids, they're duplicates of each other. But we can't know for certain that 1, 2, 3, and 4 are the same because we don't know those alleles. They're the same gene, they're not necessarily the same allele, and we just don't have that information. Does that make a little more sense? Cool. Now, life cycles are obviously going to have to, at least when you're talking about a sexually reproducing organism, require both mitosis and meiosis. So in multicellular organisms, meiosis is used to reproduce sexually and generate those sex cells in the first place. And then mitosis is used for growth and development. So specifically, when we're talking about those sperm and egg cells, we refer to them as germ cells, which are there and they're going to produce those gametes by meiosis. So those germ cells are like that basal form, right? Before they were ever a true egg cell or a true sperm cell, that's what they were. And then they had to be broken down into and processed into a traditional sperm versus a traditional egg cell. Now, only specialized cells called germ cells can carry out meiosis. So you're not going to have random liver cells doing it, hopefully. And human germ cells are found exclusively in the ovaries and the testes. It's the only place that they'll be found. And oftentimes they're usually there from the second you're born. Now, these germ cells are going to undergo meiosis, which is going to produce those gametes of sperm and egg cells, and these gametes ultimately do not divide. Once they're produced, they're produced. Now, again, remember that gametes are haploid, which means that they only have one copy, not two, one copy of all those genes. And meiosis reduces this chromosome number by half. That's how the actual process happens. Like you're going, mitosis, obviously, you're going to end up with the same amount of genetic material every single time. Whereas in meiosis, you're going from that diploid cell to a haploid cell. Now, these gametes are the only haploid cells in our life cycle. All other cells are diploid. And this is not always the case with every organism, especially when we talk about plants. Learning where and when a plant is going to be haploid versus diploid is pretty important. And it can determine what kind of plant it is. Now, ultimately, these gametes will fuse together via fertilization and bring their total count for chromosomes back up from, in humans, 23 to 46. And ultimately, this zygote is the first cell of a new organism. That's when from that point on, it's going to be mitosis. And again, these zygotes are diploid. They have the 2 full sets of chromosomes, 1 from each gamete. And that zygote is going to undergo mitosis, not meiosis, to develop into adults. All of these body cells are going to be genetically identical to the zygote. So in theory, if you were wanting to alter the genetic code of something, that's when you want to do it. Now, haploid gametes give the offspring the right chromosome number. Because what can happen possibly in some circumstances, particularly if meiosis goes wrong, is you may end up with extra chromosomes. And depending on what chromosome is extra and where exactly it shows up, it can cause massive damages to the overall functional health of that organism. So here in humans, we have 46 total chromosomes in 3 different pairs. So each sperm and egg cell will be carrying 23 chromosomes by itself because it's haploid, right? And then when they fuse together, that's going to give you a zygote with 46 chromosomes, just like the parents. And I know I keep on kind of repeating the same general information, but I promise you it'll be useful. All right, question 2 here. If an adult goat has 60 chromosomes in its body cells, its somatic cells, right? Why don't baby goats have 120 chromosomes per body cell, right? It's a pretty straightforward answer. I don't think we need to wait for y'all's answers, honestly. Baby goats do not have the body cells, only zygote cells. That's not correct, right? Somatic cells are only used for reproduction. Again, that's not correct because the somatic cell isn't a gamete cell, right? It's not those germ cells that we were talking about. Goat sperm and egg cells have only 30 chromosomes each. Probably the correct answer there, right? Because again, you're going from diploid to haploid and then you're fusing it together. And finally, baby goats undergo meiosis instead of mitosis. That's not correct. So it's clearly C, right? Just based off of what we've been talking about. Now, meiosis is extremely similar to mitosis. In fact, it's basically just the same general process, just a little bit of an extra step. So both are forms of cell division with similar phases. You're going to have an interphase, which occurs before the cell divides. And during division, the cell is going to go through prophase, metaphase, anaphase, and telophase. And finally, cytokinesis is going to take place at the end of cell division. So in other words, basically, you're going to have all these different things lining up. They're going to be pulled apart inside of the cell. You're going to recreate the new nucleus, and then you're going to separate things off into 2 specific cell membranes. Now, in meiosis, you have 2 cell divisions, not just 1. So meiosis produces 4 new cells, each having half of the chromosome from a single parent germ cell. Now, the meiosis is going to also help to shuffle genetic information, so each new cell receives a unique mixture of all the alleles. So not only are you going to get alleles from, say, for instance, if you've got your mom, right? And say, for instance, she has 3 genes, 1, 2, and 3. At gene 1, you have big A, big A. At gene 2, you have big B, little b. And gene C, you have big C, big C. Well, what can happen is you can still end up with full copies of everything. So you may not end up with the same exact version of it. And this is especially noticeable like when you have multiple genes that are all, you may have like that first gene is going to come from that first set of sister chromatids. That second gene is going to come from the second set, and the first gene is going to come from that first set. And that's something we call crossing over. Now, in meiosis, DNA replicates once, but the nucleus is actually going to divide twice. So, and DNA is going to replicate before the first cell starts to divide during the S phase, just like normal mitosis. And that germ cell is going to divide into meiosis 1. So here you can see that first initial interphase here. Here's prophase for meiosis 1, metaphase for meiosis 1, anaphase. You can see how it just it's condensed there, but you can see how it just slowly breaks things apart. And ultimately, the new cells are going to divide again into meiosis two without copying that DNA again. That's how you get that, just going from single homologous pairs here down to just getting the sister chromatin. Yeah.
Why does the germ cell have the two sets of chromosomes? Why does it have four?
So,
In the diagram?
and we'll talk more about this as we go through the diagrams, but remember in the beginning of this initial germ cell is this way, right? And so you have these homologous chromosomes. So during meiosis one, you're separating the homologous chromosomes. Now, if it was mitosis, all four of these would line up and each one would only be giving a sister chromosome. But instead, during meiosis one, they separate out the actual homologous chromosomes from each other. Then you use that second stage of meiosis. Now that we've got them separated from each other, and now you cut down the amount of genetic material in half, that's when you're actually breaking apart those identical copies based off those sister chromosomes. Does that make a little bit more sense? This can get confusing, so it's okay if it takes a second to click. Hank Green, which I'm sure y'all are rather familiar with at this point, does a really good video explaining this too, and I'll see if I can upload it. I'm pretty sure it should be good to go. I'll just have to take a look. So during prophase one, so in meiosis, the first part of meiosis, you have your initial prophase one, right? And this is where your chromosomes are going to condense and become visible, just like in mitosis. This is also where you're going to have your spindle forms that are going to start forming and then that slowly attach to the actual chromosomes itself. Now, during phase one, instead of having all the homologous chromosomes go to the center of the cell, the homologous chromosomes are going to pair up. So if you're, say, pair 22 or pair 23, you're going to go into the center and be treated almost like you would a sister chromosome during mitosis. Now, each chromosome is going to line up next to its homolog, and that's what's going to cause the actual split. Now, during this lining up of the metaphase, where they're all kind of coming to the middle of the cell here, you'll notice that this is where you're actually going to get some of that interaction between those two chromosomes. So if you look at this one specifically, you can see where crossing over has happened, because this should be all blue, right? Look at that little bit of pink. Instead, you've got that little bit of blue on this pink one, right? That's that crossing over, where you're taking basically an allele that would have been on that other chromosome, they got swapped over by accident. That's what also can help to kind of shuffle the deck and give you a lot more randomization during the sexual reproduction process. Now, during anaphase one, you're going to have separate spindles that are going to separate those homologous chromosomes, not the sister chromatids. And again, that's why I was trying to make it very clear. Homologous chromosomes, sister chromatids, they are very different things. And make sure you keep those straight in your head. And ultimately, during telophase one, those chromosomes are going to move to the opposite sides of the cell, and you're going to have the new nucleus is formed. And as a result, they're going to be now considered haploid cells, right? They only have one copy of that chromosome, not the two that you're normally associated with, like mitosis. But we're not done here. So in order to, cut down and not just have two versions of the exact same gene, right? We need to now break apart those sister chromatids. So at the end of cytokinesis one, you've got that actual cell splitting into two new cells. And then that's going to trigger into meiosis two, which is where, like mitosis, there's now going to be no DNA replication from this point on. So unlike mitosis, it's not something that's going to happen. And basically, this is the point where you're actually going to break apart the sister chromatids. So you end up with from one single germ cell that was diploid, you're going to end up with 4 haploid cells. Now, remember these germ cells have 2 sets of 2 chromosomes, and you have to get them down, so 4 total chromosomes, and you have to get them ultimately to where each of these new egg cells or sperm cells is only going to have 2 sister chromatids. Now, ultimately, this meiosis that we talked about generates that genetic variability, because not only are you only getting half your chromosomes from a single parent, so that way you're getting all the other chromosomes from the other parent. You also have that crossing over function like I was talking about. That's what can give you this basically. It's kind of like Plinko, where it has so many different options that it can fall into after, like, as everything gets exponentially bigger, right? So it's like, you have, say, 46 chromosomes, right? And on each one of these chromosomes, you have, I don't know, 1,000 genes. And ultimately, by rapidly increasing all this information and swapping it out and kind of changing it in and out, it factorially just gets massive, how many little genetic differences you can have. That's why ultimately, even though, say you have siblings, even though your siblings are still pulling from your same genetic material, both mom and dad, it may be just completely different genes, or sorry, completely different alleles for the same genes. Here's a great example of this. There's a big difference between this boring, normal ball python, this solid white snake. That's about $10,000 just between those two. And this is what we call a recessive. A lot of these are recessive alleles, some of them are codominant, and we'll get more into like how to actually predict what specific things are going to show up. But understanding how this genetic variation occurs, and how we can kind of like control for it a little bit, can be extremely lucrative from a lot of different perspectives. Both from just a general fascination and science perspective, as well as if you'd like to make a little bit of money. And again, as we mentioned, this crossing over is something that really happens during prophase, or during that meiosis one and prophase, where basically when these homologous pairs pair up during that initial part, you can accidentally have these crossing overs where you have this is part of the sister chromatid, this is part of the chromatid. And when they get pulled apart during the actual separation step of that initial meiosis one, you're going to end up with one homologous chromosome with this part that originally came from here and vice versa. Does that make a little more sense? Yeah.
So does crossing over happen in both prophase 1 and metaphase 1?
It's kind of all one combined process. It doesn't really, it's kind of obnoxious, but basically when they initially line up is when they're actually going to bind to each other, but during metaphase 1 is when they're actually getting pulled apart, or right before they get pulled apart. So it just kind of depends on where exactly you draw the line. So it's not exactly a hard, this is at prophase 1. It's kind of all kind of a continuous process, but we have to define things relatively as much as we can into groupings. And again, this crossing over is going to shuffle those genes. So this set of sister chromatids, right? So you have these two homologous chromosomes, these two sister chromatids. This one's completely different now than it was originally. And it's also completely different from the one that got separated from it during that initial state of meiosis. And as a result, there's different alleles up here than where they're original. And that can completely change the entire complexity of like what's going to show up in this organism. Now keep in mind too, a lot of these genes and all the allele swapping and all that stuff that occurs is completely independent usually. There are very few genes out there that are tied to a specific gene. So if you have an allele at site A, an allele at site B, more often than not, they're going to be completely independently assorted from each other. So whether, and that can kind of complicate things a lot when it comes to just trying to track what genetics are going to show up where. And it's kind of developed a lot of statistics and frequency metrics and all this kind of fun stuff that we've had to use to figure out exactly when a specific allele is going to show up where based off of what the parents are giving it. Yeah. No. Unfortunately, none of genetics is very simple. Something you'll find a lot. So keep in mind too, right? Basically, the only perfect way to guarantee you're going to have the exact same combination show up in the offspring is if you line group. So say, for instance, going back to that ball python example, if I want an albino ball python, and I'm going to have to basically read albino ball python to albino python. And the reason for that is it's homozygous recessive, which means that basically, if you have a big A and a big A, or a big A and a little a, both of those are going to show you a normal ball python. So you need both little a's, right? So in order for that to work, you have to basically be getting both little a's from parents. And you can have some heterozygous parents, but you still would only get like 50% normal and 50% albino. You pretty much have to breed two albinos together to get the same albino. And we'll talk more about all of that. That all comes from what we call, it was developed by Gregor Mendel in like the 1600s, before we even knew what DNA was, right? And all of that, like matching and all this kind of fun stuff was developed back then. So we have a general idea of how you can predict certain traits showing up. But again, it gets more and more complicated depending on how many traits you include. Because obviously when you're talking about a single trait that has, say, 2 alleles, right? For a single gene, a big A and a little a, or whatever. That's a pretty straightforward, you can usually figure out it's like, depending on the circumstances, you have somewhere between a 0% chance and 100% chance of having a specific outcome, and it's broken down into 25, 50, 75. But then if you add additional genes or additional alleles, it makes these tables to figure out exactly what's going to show up where massive, and you can have potentially upwards. What started as like a simple 2 particular options for a single gene, if you add another gene, it becomes 4 particular options, right? If you add another gene, that becomes 8. If you add another gene, it's 16. It increases exponentially depending on what you're looking at. And that's what gives, sexual reproduction such power. Because you're ultimately constantly reshuffling all of these alleles around for specific genes. Even though they might be the same exact genes, how they're coded for is completely different. And as a result, maybe that organism is better at living in a particular place or location. Now, again, this independent assortment is going to produce many of these very odd chromosome combinations. Ultimately, you're going to get a germ cell with 3 pairs of chromosomes. They're going to have 4 possible arrangements during metaphase 1, yielding 8 possible different gametes. And again, this is all done based off exponentially growing probability with statistics. I'm not going to expect you all to know all this, the math behind it. Just know the general process that the more genes you look at, the more alleles you look at, the more chances are that you can have much, much higher numbers of different possibilities. An additional aspect of this too is you're also going to have randomization and fertilization. You're talking about on a typical, I think for biological human males, it's somewhere in the neighborhood of like 1 million sperm are released all at once. Now, a number of those are going to get degraded. Again, remember, it's one of the reasons why it's a lot more difficult to make male birth control, right? Because you've got 1 million things being shot out of a cannon versus a single thing you can block all at once. And again, you do your best, but it is what it is. There are some hormonal ways that potentially may be helping with this, but it's just not as effective from what we've seen so far. But getting back to what I was talking about. So you've got 1 million different possibilities, right, of one particular combination of genes and alleles on that one specific haploid cell. And ultimately, it's going to ride to that egg cell. And one of those million, with its millionth version of that particular combination, is going to give that ultimate combination for that zygote. So it's really important to talk about like how just random and variable all this different things are, because that's ultimately leads to this crazy amount of diversity in our genes. Now, one thing I do want to mention is there's obviously times where there are individuals that look almost exactly alike or are exactly alike from a genetic perspective. And usually this happens through something called monozygotic twins. These are the ones that are genetically identical. And what happens during this process is that zygote is going to split itself. And instead of just continuing through mitosis and rapidly dividing, it becomes 2 sets of cells that are now rapidly divided through mitosis. And so that's what causes an actual identical twin. So it's not before fertilization, it's after. Whereas in dizygotic twins, which basically means you have 2 different copies of genes, essentially, you have fraternal twins, is probably the best way to put it. You have 2 sperm cells fertilizing 2 separate egg cells during the same pregnancy. And so that's why you can have male and female or different looks, even though they're still considered twins. This is fairly rare, but it's still more common than monozygotic twins. And the reason for that is especially nowadays with how they do particularly a lot of fertility treatments, the increased levels of hormones and things can cause excess releases of eggs. Excess probably isn't the right word for it, but like an additional increase of eggs that you wouldn't normally have in those particular circumstances. And it's not like there's a lack of sperm cells out there. So one more quick question here. Many sets of triplets consist of a pair and spare. This is where you have 2 identical babies and 1 non-identical baby, producing an arrangement that requires how many sperm and how many egg cells? Yeah, it's A, right? Because not in this particular circumstance, you have 1 set of dizygotic twins and 1 set of monozygotic twins. So one set of fraternal, one set of paternal, or, and as a result, basically you have 2 eggs and 2 sperm cells. That's why it's A. And again, remember that mitosis and meiosis have very different functions. Mitosis adds and replaces identical cells, whereas meiosis is strictly there just to produce those haploid gametes and unique DNA. But however, while they are for different purposes, they have extremely similar processes. So you might want to take some time as you're studying for this part of the test, make sure you're kind of making sure you know exactly what happens in one versus what happens in the other. So obviously they both have 2 cell divisions, crossing over, independent assortment, all that kind of stuff is what makes meiosis different. But ultimately the overall process is exactly the same. Now errors can occur in meiosis, as we've kind of been mentioning and alluding to. So if a chromosome fails to separate properly, so if you've got these 2 homologous chromosomes and one extra one doesn't break off properly like you expect, you're going to end up with the wrong number of chromosomes, and this can lead to abnormalities in the offspring. This is specifically called non-disjunction, and it's a result of some classic mental and cognitive health disorders that we often see. Specifically, trisomy 21 is also known as Down syndrome. And basically what happens is during trisomy 21, you end up with an extra copy of that 21st or that 21st chromosome, and that's what's actually causing problems. This gets really complicated when you get outside of humans, though. And the reason I mention that is there's a lot of circumstances where, for whatever reason, humans are a little bit too complicated and our genome doesn't quite work right with having a little too few or too less, or too many. But there are organisms out there, particularly amphibians, fish, plants, that there's no discernible difference between, say, a triploid or a tetraploid versus a diploid organism. In fact, there's species of tree frogs in North America. Like you literally go about another 100 miles up the road here going north, and you'll start finding them again. That are called gray tree frogs. But what's really fascinating is at some point during their evolution, some sort of non-disjunction happened across the entirety of the gamete cell. And as a result, you have a diploid version of this population, and you have a tetraploid. In other words, you have a bunch of individuals that only have 2 copies of the entire genome of the organism, and then an entire separate population that has 4 copies. Because at some point, non-disjunction failed or happened, and ultimately resulting in a failed meiosis, and they were able to keep on living. And oftentimes what you see is that, particularly even in humans too, for this matter, if you have 4 copies, it's a lot better than having 3. And basically that even number ratio helps to kind of predict that. And what's kind of cool is we can actually use this stuff to our advantage. So for instance, there's a species of fish that we use often as a kind of a cleanup crew to kill off invasive plants. It's called grass carp. And what you can do is through radiation, you can damage the gamete cells of these organisms and make them triploid. So where they now have 3 copies of their entire genome. They're perfectly healthy fish, other than the fact that they can't reproduce. So if you're trying to control how many grass carp you're putting out on the landscape to control an invasive species with an invasive species, by making them triploid so they can't reproduce on their own, you're basically not having to worry about coming in and fixing the problem of introducing an invasive species to kill the invasive plants. It's a really useful process. It's kind of cool. And like I mentioned, non-destruction causes Down syndrome in humans and actually a couple other organisms as well. And again, this is that extra copy of that chromosome at 21, occasionally at 14 as well. And that's where you get that trisomy, those triple kit versions of that same chromosome. Now, non-disjunction is most likely to happen during anaphase 1 during meiosis. So remember, anaphase is 1. So if we think back to prophase, metaphase, anaphase, right? Prophase is when they're kind of being bound up and leaving the nucleus. Metaphase is when they're lining up in the middle of the cell. And anaphase is when they're being pulled apart from each other. So during that process is when you're actually going to get that non-disjunction. Basically, all that means is instead of your typical, let's just look at the specific chromosome here. We start with 2 homologous chromosomes with 2 sister chromosomes. Now, during this during that initial step of meiosis, you're going from 1 cell to 2 cells, You're gonna end up with all the homologous chromosomes in one and none in the other. That's gonna result in basically these two final ones having no genetic material for that specific chromosome, versus this one now you end up with super extra copies in both of those. Now you can also have non-disjunction in anaphase 2, which where instead of, say for instance, you get to this perspective, right? Not actually show it. Ultimately, what's going to happen is you can also have this in anaphase 2. So just like your typical mitosis, basically what happens is during that anaphase 2, when those two sister chromatids are pulling apart, one ends up on the wrong side of the cell, and they both stay together instead of both being separated like they should. And ultimately, it just kind of depends on how things are going to work out. You can end up with a zygote that looks totally normal, a zygote that has one extra chromosome, or a zygote that's missing one extra chromosome. Now, more often than not, non-disjunction is lethal. So those extra chromosomes tend to create defects, especially in particular genes. So if you have an extra copy of, say, I think it's chromosome 1 or 2, which are especially your largest two chromosomes, they have a lot of genes that code for very specific biological processes like that underpin the entirety of like cellular respiration. And by having that extra copy, it totally throws everything off. And usually that organism cannot function, and it usually results in miscarriage. And non-disjunction is usually what we see that can cause some of the things like Klinefelter syndrome or Jacobs syndrome, where in that last chromosome, right? That last pair of chromosomes, you have your X and your Y chromosome or your X and X chromosome. What happens with these specific circumstances is you end up with an extra copy of one or the other, or just a single like copy of it by itself. And these can result in various different aspects of changing how biologically things happen, and can change how you develop your secondary sexual sex characteristics. So specifically in Turner syndrome, a female is born with only one X chromosome. This disorder could arise as a result of non-disjunction, usually in either the sperm cell, the egg cell, or both. And because in this particular circumstance, we can't determine, so remember, right, if you're pulling alleles from your parents, right? If you're from the biological father, you're getting an X and a Y, and from the biological mother, you're getting an X and an X. Now, in theory, that single X could come from either parent. So we can't really definitively say if it's the male or the biological female that contributed that less than desirable cell into that process of making that zygote. Now, all this stuff can have some really interesting impacts for evolution and ecology of particular organisms. For instance, evolving parasites are going to select for sex in worms. So in other words, there are certain parasites going to show up in here that are going to completely change how this worm is from a biological perspective, either biological male or biological female. So why do animals spend so much time and energy sexually reproducing? So they looked at this worm that can either reproduce sexually or asexually to kind of just try to start answering that question of what causes you to shift from primarily reproducing sexually to primarily reproducing asexually. Ultimately, these worms are exposed to either dead or live infectious bacteria to see if the dangerous infection affected that worm reproduction. And here's what they were able to identify. So over generations, the percent of worms that were reproducing sexually dramatically increased. In other words, as they were exposed to more and more and more of those bacterial colonies over time, it led to these worms becoming more and more sexually selective instead of being asexual. And there's a lot of potential reasons for that, right? As we've been kind of hitting on, the grand theme of today is diversity, right? Genetic diversity leads to incredible things. And so if you want to be able to respond to a pathogen, maybe you should have some genetic diversity when it comes to your immune genes. Makes sense, right? And because those genetic variable offspring were better able to survive those bacterial infections because they had new combinations of all those genes instead of just being an exact copy of their parent, they were the ones that we're going to be selected more for. All right. Like I said, today's a short lecture. So do remember quiz 8 is due on Sunday, and Connecting with Biology 2 is due on the 1st.